When you imagine a paleontologist, you might picture someone carefully brushing dirt off a skeleton with a paintbrush and covering one of history's greatest treasures, a dinosaur. But for every fossil that makes it into a museum and onto our Instagram feeds, there are dozens more that remain in the ground, out of reach or too fragile to remove without the right tools. But what if we could bring the lab to the fossils instead of the other way around? Hi, it's me, and this is my channel. And today, we're talking about how advanced imaging techniques are changing the way paleontologists search for, study, and display ancient fossils. In the past few decades, technology has made some incredible leaps, and those leaps have even helped us take a peek inside fossils without having to break them open, which is great, because sometimes the fossils themselves aren't so much fossils as they are piles of rocks, like this 318-million-year-old coral fossil found in Belgium in 2022. It was extremely well preserved, but incredibly dense and tough to cut through. So researchers used synchrotron radiation to scan the fossil and reveal its shape, structure, and mineral composition. A synchrotron is basically a particle accelerator that uses magnets to steer a beam of light around in a circle. When that beam exits the machine, it's super bright and can be focused to a small enough point that it's essentially a tiny scalpel that can slice through objects without damaging them. In this case, the scientists were able to create a map of the coral fossil down to 1/1,000,000th of a second, which revealed its shape and texture without destroying the fossil itself. And while it may seem like a strange technique to use on something as seemingly simple as a coral, it's actually a really good fit. Corals are made up of calcium carbonate, which interacts with light in a predictable way. This means that scientists know exactly how the fossil should appear when they put it under their makeshift light scalpel, making it easier to piece together and analyze. But not all fossils are so easy to work with. Take Basilosaurus, a 47-foot ancient whale monster that terrorized the oceans in the late Eocene epoch, around 35 million years ago. It had a huge skull, no dorsal fin, and an elongated paddle-shaped tail for steering underwater. Basically, it was like a sperm whale crossed with a crocodile, and would have been a nightmare to swim into. But despite how terrifying this beast was, we still don't know what it looked like because its skull is so darn big. Researchers haven't been able to find one in good condition in more than 100 years. Even if they did, they would have a hard time studying it since it would be over 6 feet long and fossilized solid as a rock. But luckily for us, and for anyone else who's afraid of giant crocodile whales, we don't need to unearth the complete skeleton to learn about this creature. Thanks to a technique called micro-computed tomography or micro-CT scanning. Micro-CT scans are kind of like regular CT scans, except they're much higher resolution, about 1,000 times higher than a human CT. So instead of looking at bones, they can see cells. And it doesn't just show skeletons, but any kind of object you put inside the scanner. You can also get a sense of the material properties of a fossil from these scans by looking at how the X-rays pass through different parts of the fossil. In 2022, scientists in Argentina scanned a small fragment from a Basilosaurus skull they found in 1985. They wanted to know if the bone was compact or spongy tissue and how vascularized it was. That is, how many blood vessels ran through it. It turns out that the outer layer of the bone was compact and strong, probably providing protection against predators, while the inner part was full of holes and lightweight, which would have helped the Basilosaurus float. Researchers are now using this method to study other Basilosaurus fossils from around the world, even though they haven't been able to move them from their permanent homes in museums. This lets them compare anatomical features between different individuals and shed light on the evolution of giant crocodile whales everywhere. But there are even more exciting things on the horizon. Scientists are now working on ways to improve micro-CT scans by adding color to them. Regular CT scans can distinguish between different densities, which appear in different shades of gray. But color scans can do the same thing and provide additional information about the chemical composition of an object. In 2022, researchers in Germany developed a new method to add color to micro-CT scans. They scanned a range of fossil samples, including a trilobite, a mastodon tooth, and a dinosaur bone, and compared the results with samples that had been chemically stained. The colors came out pretty accurate, which means scientists might soon be able to tell whether a fossil is full of iron, for example, without having to physically stain it. This could help researchers quickly identify fossils that contain useful minerals, and therefore might have formed in certain environments or from animals that lived in those environments. It might even help solve crimes, like the mystery surrounding Sue, a Tyrannosaurus rex that was excavated in 1990 and donated to the Field Museum in Chicago. She was such a celebrity that the museum had to hire security to stop people from stealing her bones. But in 2010, somebody stole her favorite snack, a 90-million-year-old coprolite that was literally $500,000. The coprolite was eventually recovered, and DNA analysis confirmed that it really was Sue's. But if a thief ever stole another fossil poop again, it would be harder to prove that it belonged to the Dino Diva. Microquartz scans can already help identify fossils that look similar, but are actually different species. So it's possible that in the future, these scans could be used to track the unique mineral composition of individual fossils, which would make it easier to reunite them with their long-lost friends. Microquartz scans are even helping paleontologists study the insides of fossils. Normally, researchers can only look at the outside of a fossil, but recently, they've started using a technique called virtual endoscopy to see inside the fossilized remains of dinosaurs and other extinct animals. Virtual endoscopy works the same way that regular endoscopy does, by inserting a scope with a camera attached to it into a fossil. But instead of poking a tube through an animal while it's still alive, researchers use microquartz scans to create a 3D model of the fossil and then virtually insert the camera. With this technique, scientists can see inside fossils without breaking them, which is helpful for studying delicate structures like blood vessels or the digestive system of a dinosaur. In fact, virtual endoscopy was recently used to study the fossilized poop of a T-Rex named Scotty, aka the Scotty 3D poop emoji. The results showed that Scotty had a gut microbiome similar to that of birds and crocodiles, but unlike living relatives of T-Rex, the team couldn't find evidence of Scotty having a specialized diet or gastric acid strong enough to digest bone, which suggests that Tyrannosaurus got most of their nutrition from plant matter. The use of non-invasive techniques like microquartz scanning and virtual endoscopy is growing in paleontology, especially among younger scientists. It makes sense considering that this technology is relatively new and easy to use, even for people without special training. It also lets researchers tackle questions that would be impossible to answer any other way. Like, what was the stomach of a Basilosaurus like? What did Scotty eat? How does the brain of a fossil differ from the brain of a living animal? There are still a lot of questions we can't answer without smashing up a fossil. But thanks to high-tech, non-destructive techniques, scientists are getting better at finding answers to the ones we can. So maybe the next time you see a fossil, it won't be a dusty old skeleton in a museum. Maybe it will be a colorful 3D model floating in front of you, or a dino poop emoji with an explanation of its surprising microbial diversity. Thanks to my supporters for making it possible for everyone to learn about the amazing science happening in our world today. And thank you for watching.